Okay, so as you can see, we have the whole DNA strand taped to the board. I noticed that this DNA strand is really kind of a mess. It needs to be erased, like cleaned. So if the lab tech wants to spend some time cleaning that, it would be nice, and taking the tape off and kind of getting it in better shape. All right, so we've got our whole DNA strand on the board. The students also have small laminated copies of this thing that they can have at their table. So you want to have those out so that they don't have to come up to the board and do it, but this is a replica of what's up on the board. Okay, so they're going to have their student guide, and you're going to go ahead and just ask them to do steps 1 and 2 of their student guide. Okay, so step 1 is asking them about a promoter. It's telling them that it's the TATA box, and the terminator, which is the polyadenylation signal, which is AATAAA. And so with their partner, they're supposed to find the promoter and the terminator. They can either do it on their little laminated piece, or they can come up here and find it on here, and I'll zoom in a second and show you where that is. And that's number 1. And then number 2, it asks them if you want to make an exact copy. So remember, and this is like back up and say, this is our master recipe book. So this DNA is in the nucleus of the cell. It is the only copy that cell has, and it's been handed down from generation to generation. So it's really old, per se. Okay? So it's a really old code, and it's the only one the cell has. So I like to liken it to a really old cookbook. So if I had a cookbook that had been passed down to me from my great-great-great-great-grandmother, the question is, would I, if I wanted to make chocolate chip cookies, would I bring the cookbook out, slap it down on the table, and start making cookies on top of it? That wouldn't be a very good idea, because you could ruin the cookbook. Now I do that all the time at home, and I ruin the cookbook. So you don't want to ruin the cookbook. So the better option is to leave the cookbook in the Smithsonian, right? But I want to analyze one of these recipes, so I'm going to leave it in the Smithsonian Library. I'm going to go to the library, I'm going to open it, I'm going to make a Xerox copy of the recipe that I want, and then I'm going to leave the book in pristine condition in the Smithsonian, and I'm going to take my copy out into the kitchen, and then I can make cookies all over it, because it doesn't matter if it gets ruined, because I've got my master copy in the nucleus. So that's kind of the idea here. Okay? So students are trying to figure out how might the cell do this in a way that's efficient. How do they make a copy? And I like to remind them that cells don't have eyeballs. So it's not like they can read it and figure out and just write it down like we would. Okay, they can't read, so they're going to have to do it blind. They're going to have to feel somehow. So that's a little hint. You walk around and have students sort of hypothesize how they might make a copy of the DNA. Okay, so give them a minute to think about that. Now, depending on who your lecture person is, they may or may not have covered DNA replication yet. And so it's up to you whether, I would not mention DNA replication at this point, because they haven't really covered it in lecture, or they may have, but some haven't. So just say, you know, if you were to hypothesize a way that we might do it, because it does happen similar to DNA replication, but since they haven't had it, don't mention it. All right, so I'm going to zoom in so you can see what we're going to talk about. Good. Okay, so I'm just going to talk about the hemoglobin gene. Okay, so the hemoglobin gene, you can see it's actually traced in blue. It's in a box. If we can get the blue off, that'd be great. So the hemoglobin gene is from here to here. Okay, and emphasize to them that a gene is just a single strand sequence of DNA. A, T, C, T, C, G, T, C. It's a recipe, right? We're reading every 3 letters to make the amino acid. And so it's just one of the strands. It's not the other strand. So what they're finding on number 1 is the TATA box, which should be upstream of the gene. So you can see it here, TATAAA. This is our TATA box. That's your promoter. That's how the enzymes know where to start transcribing. And then our polyadenylation signal, our termination spot, is right here, AATAAA. So this is our terminator. Okay, it's a polyadenylation signal. That's what signals it to add the poly-A tail, which we'll learn about later. So they should have been able to identify this and this. Now, I would also emphasize to them that this is greatly underestimated here, right? That this is normally, the TATA box is normally hundreds and hundreds of bases upstream of the gene. But just for their sanity, we didn't make it that big. My sanity. And same thing with the polyadenylation signal. It's oftentimes way downstream, but we're making this more simplified. okay? All right, so if we're doing number 2, then we might ask them, okay, if we don't have eyeballs, we can't just write it down. I can't just say, I want to copy this gene, so I'm going to write A, T, G, C, A. I can't do that, okay? The cell doesn't have eyeballs. And so the easiest way for the cell to do this is to split it open and then to feel the bottom strand. So if I feel the bottom strand, if I see a T, what do I know was across from it? It was an A. If I see a C, what do I know was across from it? A G. Okay, and if they haven't, if the students are looking at you confused, like they haven't covered complementary base pairing yet, then just mention to them that in DNA, we did it at the very beginning of my class, but the A always goes with T, C always goes with G. So you might just write that up there, right? A goes with T, C goes with G. Now, in RNA, that's going to be a little different. We'll write that up in a second. All right, so we're pretending we're splitting it open, and then we're going to use this bottom strand as a template in order to know what the top strand was. Almost like an impression, right? Like a tooth impression. You do an impression of your teeth for like a retainer, then you see those dips. You know exactly what the tooth actually looked like, okay? And so that's what this is. This bottom strand is a template. And so we call the top strand the code. All right, so the top strand is the code in this case, and the bottom strand is the template. And something to remember is that, and to emphasize with students, is that we always read the coding strand 5' to 3'. And if they don't know what 5' and 3' is, you might remind them if the orientation of the DNA. I don't know how much detail the lecture people went into. You don't have to go into this detail, but if students are curious, when you have a DNA strand, DNA is made up of these 5 carbon rings, right? And then there's a phosphate group and then another one. And then the base is sticking out here, so there's an A. And then it would be paired to a T. And then over here, your DNA is going to be upside down because it's anti-parallel. Oh, that's an O. Oops, that's an O. Okay, and your phosphate group. So there's their DNA strands that are running anti-parallel. The way that we count them is we count from the inner carbon. So this is carbon number 1, 2, 3, 4, 5, and this one is carbon number 1, 2, 3, 4, 5. So this strand is running 3 to 5. And this strand is running in the opposite direction, 3 to 5. It's the orientation of the sugars. I would not go into detail about that unless students ask, unless they're curious. Okay? So we're going to erase this. Okay. So, if we're going to split it open and use the template, then we're going to complementary base pair to the template. In question 2 asks them, what do we call the opposite strand? We call it the template strand. Okay? And so, the way that we would do it is we'd say, okay, I see, and we're going to actually start at the data box. I see an A, so instead I'm going to write a T. But our carbon copy paper that we use, like making a carbon copy and impression, it is a different kind of material. It's a material that's designed to degrade a little faster. And so it is called RNA. So this is DNA. RNA is similar to this. It has A's, C's, and G's, but it does not have T. So we still have C to G, but instead of a T, RNA uses a U, a uracil. So you want to make sure to emphasize that to students. And if it gets confusing, I always tell them to write down the T's and then just erase all the T's and write a U. Because what students end up doing is they see a T and they write a U across from it. They get them all mixed up. So you just need to make sure, look, it's the same thing. We're substituting U instead of T. All right, so if I see, back here in the data box, if I see an A on the template strand, I'm going to write a U. If I see a T, I write an A, I write a U, T, I write an A, C, I write a G, G, I write a C, G, U, okay? So that, taking it from DNA language to RNA language, so we're taking it from DNA, which is this, to RNA, which is that term, we have transcribed it. Okay, just like a medical transcriptionist takes the gobbledygook the doctors say and transcribes it into good notes, that's just taking it from one language to another. Okay, so taking it from DNA to RNA, we're transcribing it. Okay? All right, so I'm going to just pause and make sure this is taping.